For today, we're going to be discussing some diagnostic microbiology. When we consider human health and the fact that microorganisms are typically at the center of a lot of problems, especially diseases that can arise, one of the things that we need are tools and able to diagnose what kinds of infections somebody may have that causes a specific disease from a variety of different samples that are taken from the human body. Many of the techniques that have been developed around this are used for the prevention, the diagnosis, or the treatment of these infectious diseases. The bottom line is that if you don't know what kind of organism that you're dealing with, you won't have an effective path forward, especially in the realm of treatment. So there are many different types of specimens that can be taken from a person who's infected and analyzed to identify or at least narrow down what type of organism they may be dealing with. We're going to be talking about a variety of different locations and different sample types and some of the methods that can be used to ascertain what kind of organism is present in those specimens. One thing to consider is that specimens have to be obtained and handled properly. So if you can imagine, we are humans that have our own commensal organisms and we are taking samples from another person, there's a high potential for contamination if things are not conducted properly. There are quite a few different factors to consider, like taking specimens from the infection site. So for example, if you had a throat infection, you'd want to make sure that you're testing from the proper areas of the throat versus just spontaneously somewhere within the oral cavity. The samples also have to be taken aseptically, so you only want to have stuff on your sterile swab from the site of infection and from that person. So you don't want to breathe on your sample, touch your sample, put it down on the bench when you're done sampling. Everything has to be done in a way that's going to ensure that as you're trying to diagnose, the only sample that you're assessing is from the infection itself. Even consider the idea of trying to draw blood several times, where if you're constantly putting a needle through skin to get into veins to draw, what you can actually do if you have several attempts is force skin microorganisms down into that sample that'll end up contaminating it. So there may look like there's something present in that blood that should not necessarily be there, but that was introduced from human error versus actually being a part of the problem. Another aspect is that the sample size has to be large enough for testing. Really, you only need a single cell, but the more that you have, the better, of course. The metabolic requirements also have to be met, so you can have either anaerobic or aerobic organisms, among a variety of other things that you'd have to consider. Because if you take a specimen that prefers anaerobic conditions and try to grow it in aerobic conditions, you might get a totally different set of bacteria that grow that you'll be trying to treat, but that aren't actually causing the problem to begin with. So you do have to consider those things and try multiple strategies with your sample to see what will actually grow. In addition, you have to consider what else is going on with that person. If they're on some kind of antibiotic regimen, if you're looking at a urine sample, you may have antibiotics in the urine that are killing bacteria that could be causing an infection internally, but might not show up in your diagnostic test type. So it's more or less putting together a lot of different pieces of a very large puzzle with a number of different techniques and strategies. When we're looking to diagnose conventionally, a lot of samples are taken and then used to inoculate agar plates or broth cultures and eventually be able to isolate the different bacteria within. So you could use things like the streak plate technique or the dilution plate method in order to see individual colonies, look at those characteristics, and then determine what they could possibly be. Another way around this is to actually do DNA sequencing or other types of molecular techniques for identification that are a little bit more rigorous versus just looking at colonies and trying to identify that way. You can look at protein assays and other things to identify enzymes and other cell receptors that might only be specific to certain species. We haven't really touched much on that, but there's a lot of diversity in terms of what types of proteins will be present in certain bacterial species, but not found in others, depending on where they live and what their metabolic preferences are, among a number of other factors. Another thing to consider is that time frame, because a lot of pathogens, while they will grow up overnight in culture, some won't. So certain bacteria like mycobacteria will grow very slowly in the laboratory, if at all. Some species can't actually be cultured in vitro. And in vitro meaning that they're not inside of a living thing. So you would be growing them in culture or on plates outside of a host. Bacteria that are very picky or fastidious or that prefer anaerobic conditions can take either days or weeks to grow. And of course, Bacteria that are intracellular, meaning that they prefer to live inside of human cells versus externally, have to be cultured in cell culture. You can't just grow them up on agar plates because those are not the native growth conditions that they prefer. So you can see no growth, but are you seeing no growth because that person doesn't have an infection? Or are you seeing no growth because you haven't met the needs of the organism that you're attempting to diagnose? In these cases, you can always look at alternative methods like DNA analysis or immunological testing, but this is always a very stepwise process where you try something, and if you see no results, then what are your next steps? Where do you go from there? You have to consider many different paths to eventually get to a definitive answer. When we're looking to grow bacteria, that cultivation can be done using a couple of different techniques, and one of them that we've talked about a lot already is isolation media. We can inoculate plates and spread them out thinly enough that we have single cells that can grow up into individual colonies. We can look at those colony characteristics and test them further if we need to. We can use general purpose media to target anything that might be in a sample taken from an infected person. We can use enriched media if we suspect that a fastidious or picky organism might be present, or we can use selective or differential media. And these again will help us to bolster the growth of a certain group of organisms that we think could be responsible, or group them based on their colony characteristics that might arise from growing in certain types of media conditions. But at the end of the day, you need to be able to grow your specimens in culture and then assess them in some way to determine their identity. There are other tests as well that can fall into one of two categories. They can provide presumptive data, where you're getting isolated microorganisms that can be placed into a category, like its genus, but you might not know the specific species. There's also confirmatory data that can provide definitive evidence for a specific species. We're going to talk about a number of different tests that either allow us to identify bacteria directly or culture and isolate them and then prepare them for further assessment down the line. You may have heard of rapid antigen testing, and this is done using something called antibodies. Antibodies are proteins that we can make synthetically in the laboratory that identify very specifically proteins. These proteins are usually going to be part of a bacterial cell's anatomy, something that they'll always have. And if it's present, if you have antibody that binds to it, you can also conjugate that antibody to something that gives us some kind of signal. So things like light or color change. And what happens is if you have a species that has the protein that you're trying to target and your antibody binds, you'll see that signal expressed in your sample. So you'll see light expression or you'll see that color change indicating that it's there. Alternatively, if you have an antibody for something that you're targeting and a species doesn't have it, you will not see that production of that byproduct, that light or that color. And what's neat about this is that you can make antibodies for any protein. And if you have a single protein, there can be hundreds of sites on a single protein that you can target with different antibodies. So at the end of the day, you're ending up with something that's extremely specific, and it's very difficult to make mistakes using antibody testing. It's really an indication of yes or no. The only trouble that you run into with this is determining a target that is specific only to the organism that you're trying to identify. So your species might produce a certain special kind of protein or an enzyme or a receptor on its cell surface that others do not. There are some great quick test kits for several species. We see these all the time for strep. If you go into the doctor's office with strep throat, they'll take a quick sample and throw that into an antibody detection assay. And right away, if you do have strep, you'll see a color change in that medium to indicate that you do have that infection. So these are really useful. They don't require external processing. Those samples can also be cultured up to verify a certain infectious agent being present. But for an initial diagnosis, they're very handy because you can see those results with the naked eye, those color changes, that light production, whatever that indicator might happen to be. You can also perform biochemical testing, where you're looking at the reactions that occur within different bacterial types. These are primarily based around metabolic reactions, because lots of different bacteria are going to have different metabolic preferences, as we've been discussing. And so you can subject your bacteria to a variety of different kinds of agar to assess what they prefer. So things like the starch test, the gelatin test, or other kinds of assays that target certain types of enzymes that are part of metabolic pathways that may be unique to different species that are pathogenic can be exploited in these cases. If you're dealing with either intracellular or extracellular bacteria, you can use a variety of microscopy methods. You can look at immune cells and bodily fluids to look for different types of infectious agents, or even in the case of something like syphilis, you can look at moving spirochetes that would be present in urine samples if that infection is available. So you can see some bacteria just through microscopic analysis of different species. And of course, there are stained preparations that you can do, like gram staining to differentiate gram negative and positive cells, acid fast stains to detect the presence of mycobacteria, and of course, fluorescence assays. Lots of these methods are fantastic, but they do have some limitations. Gram staining is great to class most microorganisms, but if you consider mycobacteria not being penetrable by gram staining, or things like spirochetes that are too thin to be observed through fixation step that would be required for that, they're going to be more difficult to identify with these methods. You can also consider that intracellular bacteria are difficult to identify this way as well. While stain will get taken up by eukaryotic cells, it's not going to have the same effect on internalized bacteria that it would on those that would be external. There's also the potential to try out some blood cultures, and this would look at the presence of bacteria in the blood. That would reveal a situation called bacteremia, where you do have presence of bacteria in the blood, which is not a common occurrence in people that are healthy, and it's usually indicative of a systemic infection. This could be leading towards a state of sepsis, for example. Septicemia is when you do have a very large, widespread infection in the body, in the blood specifically, and the movement of those different bacterial species throughout the body to different organs will cause systemic shock. In these cases, this is a result of our immune system pulling out all of its cells out of the bloodstream and into the tissues where these infections may eventually end up. If you imagine our blood volume needing to be at a specific amount to maintain our blood pressure, as soon as we have a lot of our blood cells exiting en masse body-wide to go fight infections everywhere, all of a sudden you can end up with a drop in blood pressure, which of course can result in inefficient amount of oxygenation to different organs, which can result in death over time. So if somebody does become septic, there are different treatment methods, like antibiotic treatment, if you can get people to the hospital in time. But sometimes if sepsis is allowed to go too long, it's very difficult to get people to come back from that state. When you're looking to determine whether or not people have a blood infection, you take 2 blood tubes, and then one gets incubated in aerobic conditions and one is incubated in anaerobic conditions. If there's growth, those samples can be further assessed. Unfortunately, there are some organisms on the skin that can contaminate blood samples. We did touch on this earlier. The problem with this is that while they are commensal, some of those organisms can be opportunistic and have been implicated in pretty serious conditions that affect some of the major organs, like the heart. So if skin contamination happens in your blood samples, you may start down a diagnostic path that might seem a lot more serious than it actually is. So that's just to say again that making sure that these samples are taken aseptically and properly on the first go is really important. I'll give you a couple of examples here now that can be implicated in some of these issues. Pathogenic cocci are highly implicated in many infections on the blood and the skin. These are pyogenic, meaning that they cause pus. There are 4 different genera that have the species that are responsible for these kinds of problems: Staphylococcus, Streptococcus, Enterococcus, and Neisseria. Other than pus formation, lots of these species have other enzymes that can be detrimental to human health. We'll start with Staphylococcus aureus, which is pretty versatile. This is something that's normally found on the skin and can withstand a lot of different changes in environmental conditions. But species that are pathogenic are going to have several really terrible enzymes, at least in the context of how they would affect us if they're within our own systems. Coagulase is an enzyme that clots blood and plasma. If you can imagine, you don't really want your blood being clotted unless you want it to be clotted. But a lot of these species will actually clot your blood and then hide inside of the clots that they create so that your immune system can't find them. They also produce lipases, which are going to help bacteria to colonize oily skin, or penicillinase, which is going to inactivate penicillin. They are susceptible to penicillin treatment, but they do have enzymes that can cleave that and make the medication ineffective. Exotoxins are also produced by members of this genus, and exotoxins are those that are secreted. So they're produced by the bacterial cells and then released into the external environment to affect their targets. Hemolysins are going to destroy our red blood cells. Again, not good. and leukocidins, which will damage the membranes of neutrophils and macrophages. Neutrophils and macrophages are two types of immune cells that are usually first in line to help to fight new infections. And if you have those being destroyed, of course, we have a reduced immune response, which may allow bacteria to live longer in a host and establish themselves better. The longer an infection is allowed to persist, the worse it is to treat and the more difficult it becomes to treat. Staphylococcus aureus does cause a number of diseases of the skin, and they are really the cause for pimples that we get on our bodies. You can have folliculitis, which is the inflammation of a hair follicle. You can have furuncles, that are boils that can come around from inflammation of a single hair follicle or one of your sebaceous glands. Or you can have carbuncles, which are large, very deep skin lesions that are caused by many furuncles that have formed in an area. Staphylococcus aureus is also responsible for impetigo, which you may know as a very infectious disease of the skin. This is something that actually gets spread a lot between young children that have the infection, and it's caused by an itching blister that tends to shed quite a lot. It'll ooze, it'll crust over again, and then kids, of course, are touching everything and everybody around them, so you do see that infection spread a lot in young people. If somebody does have impetigo, it's important to make sure that you're washing all of your bedding and your towels as often as possible, because it can even be spread through those means as well. A really easy test to identify whether or not a specimen does contain staphylococci is by using the coagulase test. Because they produce coagulase, if you add staphylococci to a sample of blood plasma, you do see the formation of tiny clots in that specimen. You can see in this example here, we've got a species on the left that's been added to plasma, but the solution is quite uniform, so you're not seeing any clotting or clumping. On the right here, though, when we add staphylococcus, you can see that there are little dots of particulate that have formed, and that is due to coagulase coming in and binding a bunch of blood factors together to form blood clots. So this is a really great, quick and dirty diagnostic that can tell you if staphylococcus is present in your sample. Streptococci are another group that can be classified as either commensal or pathogenic. Sometimes both, right? We can have opportunistic commensal organisms that fall into this category. There are a lot of antibiotic resistance members of this group, and some of the pathogenic species that we'll talk about include Streptococcus pyogenes, pneumoniae, and Algactiae. Streptococcus pyogenes is the most serious pathogen of this group because it has parasitic tendencies. This is the one that will cause what we call strep throat, and it'll inhabit the tissues in and around that area of the body, but can also live on the skin. Streptococcus pyogenes has a couple of different virulence factors, and virulence factors means how disease-causing are they? Diseases are characterized by their symptoms. So if something is very virulent, it causes a lot of problems or negative effects in the host. Streptolysins are a type of factor that's produced here, and these can cause the destruction of red blood cells as well as many other kinds of tissues, like liver and heart muscle, and they're also pyogenic. This can cause a bright red rash that can appear all over the body and can also induce a fever state by acting on the temperature regulatory centers of the body. There are a lot of very potent enzymes that are secreted by this species, like streptokinase, hyaluronidase, and streptodornase. You can see something like streptokinase is going to help to get past some of our immune responses mechanisms for containing, because our immune system will produce blood clots to help to wall off infections from spreading. And these bacteria know how to just break through those barriers. Hyaluronidase can break down connective tissue. Streptodornase hydrolyzes DNA. So in all of these cases, these are very detrimental to the host. And you might be wondering why we worry about these widespread body effects when we're talking about a disease-causing agent that's usually going to stay in one tissue. Most of the time, strep throat does stay put, but on occasion, those specimens can get into the bloodstream and become a systemic infection, which is when all of the problems that we've just discussed become so problematic. We do tend to see it primarily presenting like this in the tonsils, where you do see a lot of pus forming on the tonsils, which results in a lot of pain. The pain comes from the inflammation of the tissue or the swelling. While this is a result of our immune response doing its work, this is one of the more painful throat infections that you can get. Scarlet fever is an example of one of the diseases that can occur when Streptococcus pyogenes is going to spread systemically or throughout the body. It tends to be a rare complication of the throat infection, but as it spreads throughout the body, through the bloodstream, it gets into different tissues, resulting in that diffuse rash that we've talked about, and is very dangerous if it's left untreated. There are some characteristic identifiers for this disease, so it's easy to spot, obviously, but one of them is the red tongue. This is something that doesn't really present in a lot of other cases, but in terms of a systemic strep infection, that will show up most of the time. When we're looking to identify Streptococcus in the laboratory, one of the things that we can do is test its sensitivity to bacitracin. Streptococcal species are extremely sensitive to bacitracin at low concentrations. And bacitracin is one of many kinds of medications that we develop to target and destroy bacteria. It's kind of like an antibiotic because it is used in cell-to-cell warfare outside of our practices with it. Another thing that we can do is look at Streptococcus species on blood agar. Because of that hemolytic activity, or the destruction of red blood cells, if we culture Streptococcus in this way, and we see the removal of the red pigmentation that's usually there because of the red blood cells, we know that those cells do kill red blood cells and destroy them, which would imply that they're part of this genus. You can see on the left here that zone of inhibition around bacitracin, which has been applied to cells grown up on blood agar. And over here on blood agar, you can see those zones of clearing. Different streptococcal species can have varying degrees of hemolytic activity, and we'll talk about that more in the laboratory. Another type of specimen that can be taken from the human body are urine cultures. These infections typically arise from commensal organisms gaining access to the urinary tract. So they're being introduced into the internal aspects of the body through external methods. Infections of the genitourinary tract typically occur because of E. coli, which is an enteric bacterium, but that can easily contaminate other parts of that area and can in turn produce infection. Bacteriuria is the presence of bacteria in the urine, and this can be identified simply through microscopic analysis of urine. You put some urine on a slide and have a look, and if bacterial cells are present, then you can determine that there is an infection there, because they're not typically supposed to be there at all. Urine is actually not sterile. A lot of people will insist that urine is sterile, but it actually is not, especially as it comes out of the body. As it passes out of the urethra, it has a tendency to pick up some of the bacteria on that external tissue. One of the ways that we assess urine is through a dipstick test, and this will help us to assess a bunch of different things at once. You can look at different types of metabolic intermediates and whether or not they should actually be there. A lot of different diseases can be determined this way based on how our urine is being produced and what deficiencies that we actually can have based on a disease or even some kind of somatic dysfunction. If we have dysfunctioning organs, we will see problems in the urine and the balances of the chemicals that are usually there in a healthy individual. Fecal samples are also really interesting to assess, but they do come with their own trials. Unfortunately, when we have to store fecal samples for a long period of time, they can become more acidic, and acid levels can destroy most enteric bacteria. We've also kind of touched on the fact that the colon tends to be more alkaline in nature, so a swing down in pH can cause a pretty detrimental effect to those organisms. There are situations where samples will contain pus or blood or other factors, so selective media is usually used to identify the species within. So things like that, eosin methylene blue agar or even McConkey can be used in these cases. If someone is sick because they've been traveling and perhaps drinking some contaminated water, other species can even be isolated from the water itself and used to compare to samples that are taken from a sick person. Scaling it up a little bit to eukaryotic infections for parasites, most of them will produce what are called cysts, and those can readily be observed in fecal samples and identified. You can see in this microscopic specimen here, these large entities are actually Giardia cysts that will show up in people that have fecal oral contamination from a variety of different sources. When you want to look at cultures from wounds or abscesses, these are normally bacteria that are opportunistic. So they have lived in and on us and then have turned for whatever reason with an opportunity to cause infection. The best way to assess purulent regions that are pus-filled is to actually aspirate with a syringe. If you choose to disrupt something that is swollen and full of pus, you run the risk of introducing it to the surface of the skin and contaminating it. So putting a needle in and taking some material out is much better. A lot of the species that live inside of the skin in these closed off areas are anaerobic, so samples do have to be grown up in both of those conditions, being aerobic or anaerobic, to make sure that whatever's causing the infection actually grows up in large enough numbers to be identified. We can also assess a lot of genital specimens that may arise from sexually transmitted infection. Gonorrhea is one such example that is caused by Neisseria gonorrhoeae. This disease will present differently in males and females. In males, we have purulent urethral discharge that can be sampled and cultured for identification. But in women, it's not so visible. One great way to determine whether or not females will have this infection is to catch early morning urine. Because if the infection is present, it will build up overnight. And when you expel that first bit of urine in the morning, there will be a higher load of these bacteria that can then be assessed. Neisseria gonorrhoeae is a gram-negative diplococcus, and there is no other commensal organism that have this particular arrangement, so they're very easy to spot compared to other things that may be present. And you can see some examples of some diplococci. Other methods can be used to verify whether or not gonorrhea are present, and chocolate agar contains heat-lysed blood cells that appears dark brown, so not initially as tasty as you might have been thinking. There's another kind of selective media called modified thiomartin or MTM media that will contain a ton of different antibiotics that will affect most organisms but not Neisseria. So when you grow these species up on this plate, if gonorrhea is present, it will be there. Other things should be killed while gonorrhea will persist. After you manage to isolate bacteria using these methods, we have to subculture to measure some kinds of biochemical reactions. Because again, if you're looking at bacteria, they're all going to have their own unique metabolic pathways, which come with their own specific set of enzymes. One such example is eosin methylene blue agar, which is selective and differential, and it will help to support the growth of bacteria that are gram-negative and will also identify bacteria that ferment lactose. And this is a common feature of many bacteria that are found in the enteric regions of the body. This here shows an example of a culture grown up on this medium that produces that metallic sheen as a result of that lactose fermentation. Other tests, like the phenol red test, can be used to look at sugar fermentation. That, along with the production of gases that are measured using a Durham tube, which can be observed here inside of these test tubes, can be used to assess the metabolic preferences of a species and also whether or not they will ferment different sugars when they are cultured in the laboratory setting. A color change from red to yellow will indicate the production of acids, which are typically a byproduct from sugar fermentation. And if any gas is produced and collected in a Durham tube, that is also indicative of fermentation moving forward, because as we know, the gas produced during fermentation is carbon dioxide. So if you do see that little bubble appear, that would also indicate that fermentation is occurring. The EnteroTube 2 test is a phenomenal tool that allows us to identify the metabolic properties of a bacterium with many different media contained in one test. Each one of these individual wells, labeled 1 through 12, have a different type of detection agent in them, and depending on what bacteria you introduce, they will either grow well on each of those 12 media or not. The different combinations that can result from that will give you some insight into which species that you have. So if you look here at the uninoculated control, these color changes that we observe with different species indicate use of that medium. And you'll notice that none of these are alike because they're all different species that use different metabolic pathways. So this test is super neat and extremely useful, but because it does so much, it's also fairly expensive to use. Immunological tests are another way for us to be able to see whether or not an infection is present. When we are infected with a pathogen, most of the time our body is going to produce antibodies against it. Antibodies are produced by B cells, which are a type of white blood cell that are very active in our immune response. Antibodies usually work by attaching to pathogens, which tells our body that they are foreign and need to be destroyed. They not only bind to the pathogens themselves, but can sometimes also bind to their toxins. By binding to a toxin, it can prevent that from being able to affect its target. So we can look for the presence of antibodies against certain pathogens within a patient, or on the commercial level, we can produce antibodies that can detect those infections and then test for them in clinical samples, as we discussed earlier. Antibodies can be made against anything. So, if you can induce an immune response in an organism, you can make antibodies. What you need for this to be able to occur is for the entity to be considered non-self. So, for example, if we were going to make antibodies against a certain pathogen, if we injected that pathogen into a living organism, and in this example here, they're using a mouse, if that pathogen is foreign to that mouse is going to form an immune response against it. So it's going to raise a bunch of cells that are going to produce antibodies that are going to detect that foreign agent and bind to it. So in the laboratory, what we can do is isolate the cells from that mouse that react against that particular pathogen, and we do a fusion with cells that are more long-lived. A primary cell line, like spleen cells that are going to be producing antibody inside of a host, won't live very long in culture. But because we want to be able to produce those antibodies on the long term, we would hybridize them with cells that have a longer shelf life, so to speak. This is typically a cancer cell of some kind that does not really regulate or obey its regulatory mechanisms and can divide for many more generations than a normal cell would. Once the hybridization has taken place, we essentially have created an immortalized cell that will produce this antibody for a very long time. And then we can grow those up in culture. And then from those cells, we can isolate the antibodies that we want and produce those on a mass scale. Those are the antibodies that we can then use in a clinical setting to detect the same pathogen that was initially injected into that organism to begin with. So this is an extremely useful tool to, again, definitively identify small specific features of bacteria and definitively identify them from a bunch of others that might be present. So apart from using them for detection, antibodies can actually be used as a treatment, because if we have antibodies that we know are specific for a toxin that gets secreted by a pathogen, we can inject those antibodies into a person, and then they will come around and actually bind to the exotoxins to prevent them from being functional. This process is called neutralization, because those toxic effects of those toxins are actually negated when an antibody is bound. They can't usually reach their targets if they are inhibited by this huge other protein that's conjugated to them. I talked about light emittance a while ago being a way of seeing whether or not a certain feature is present using antibody detection, and this is kind of how this works. So if we have a bacterium that has the antigen or the feature that we're looking to target on its surface, when we add antibodies to it, they'll bind to the surface of those cells. These antibodies, in this case, are labeled with a fluorescent dye, and because they're stuck to the surface of these cells, they don't get washed away during many of the other steps in the assay. So when we look at these cells under the microscope, if we see cells that are fluorescing, it means that that target that we did target specifically with antibody is present. If we were to do this process and see no fluorescence at the end of the day, once we get to the microscope step, that would mean that the target isn't present. So the pathogen that we're trying to see if it's there would not be there in that case. The same kinds of principles of yes or no, is it there or not, can be applied to this other test called enzyme-linked immunosorbent assays, or ELISAs. These are another antibody assay that can be used to see whether or not a certain pathogen is present in a sample. They're actually used widely in testing for HIV particles. And if HIV particles are present in a specimen, we're going to see that color change that's shown here in this plate. You might be saying, well, how much do I need to know about these tests? What I'd like you to be able to know from these is which ones use antibodies and which ones don't. You don't have to know most of the details or how they work or the minutiae of them, but rather which ones of these tests use antibodies and which do not. Radioimmunoassays are another type of test that will use radioactivity instead of enzymes. The problem is that sometimes when we're growing bacteria up to culture and to test, some of the factors that we use in those media will inhibit the enzymes that cause those color changes or that production of light. So if we're using a radioactive indicator that's not reliant on enzyme activity, we get a special amount of sensitivity that cannot be achieved by other means and can be used in situations where our enzymes may be affected. But these again give you that yes or no, is it present or not, based on whether or not those antibodies will bind to the sample if it's there. Western blotting is another procedure that uses antibodies that can be used to detect the presence of antigens, but this takes it down from the level of the cell to more of a protein emulsion. So you would kill the cells, take out all of their proteins, and test them that way instead. In these cases, once you've applied these proteins and separated them out using gel electrophoresis, you can transfer them onto what's called a membrane. And that membrane is what you're going to treat with those antibodies. So if your target protein is present and you add antibodies to your membrane, it'll show up in your Western blot, but if it's not there, it won't. So these are all a very similar set of methods that use the same principles, but can be used in different ways in different times depending on what is required or even what kind of laboratory you are. You might not have the means to do radioimmunoassays, but you could do Western blotting or you could do ELISA. All of them tell you the same thing, yes or no, is an infectious agent there or not, but these are a bunch of different ways that bring you to that same conclusion. Nucleic acid tests are another great diagnostic method that can be used because they are very easily isolated from infected tissues, and each bacterial genome is unique. So if you can take that genome and effectively identify it, that is another great way to see whether or not a certain pathogen is infecting a person that you're trying to test for. If you only have a little bit of DNA, another great feature is that DNA can be amplified. We can do that using polymerase chain reaction, or PCR, where we can take a specific sequence of DNA and produce more of it so that we have a lot to test with. And it's extremely stable. So if you do happen to freeze and thaw it a bunch, it's not going to degrade over time. Proteins are far less stable. So if you're trying to look for them and test for them, they don't tend to last as long on the shelf. DNA testing does not use antibodies, and it has a lot of advantages if you have the means to test for it. Not all laboratories are going to have that degree of specialty. Nucleic acid hybridization is a very important clinical method which will detect specific DNA sequences that are associated with organisms. What you do here is take your DNA from your sample and you add a probe that has a reporter on it. This reporter can be a number of different ones. It can be, again, something that produces light or color, or even in some cases, radioactivity. But either way, it's going to tell you whether or not it's bound to the correct sequence. So if you add your probe to your sample and it binds there, that means that that bacterium with that specific sequence in its genetic makeup is present. If it's not, if it does not bind, then you will not see that reporter activity. So again, a non-antibody mechanism that allows us to see whether or not a certain type of genetic sequence is present, which would of course tell us the identity of an infectious agent. And as I mentioned, we can use polymerase chain reaction to produce more of that genetic material if it's required. And we won't spend too much time talking about PCR because we've touched on that a lot in other courses, but what you're doing is replicating the DNA synthetically to produce more of it. Viral detection is also a useful method, but because viruses tend to be intracellular and obligate parasites, they have to be grown inside of a human host. To grow viruses up in the laboratory, we have to grow them up in cell culture, and we can do that in any kind of cell line. Depending on the purpose of growing up viruses, we might grow them up in immortalized cell lines like we talked about, where those cells will be able to divide more times before they die than normal cells. Or you can grow them up in that primary cell line, which would just be like taking a sample from normal tissue and growing it up in the laboratory. Those have a much more limited number of times that they can divide before they will senesce. Once viruses have grown up in cell culture to high enough numbers, they can be easily detected through electron microscopy. I mentioned a long while back that most viruses have a very specific and unique physical appearance, and based on that can be very easily identified without too much need for further testing. Though if you'd like to verify, you can also use any of those immunological methods that we discussed or even the DNA hybridization technique. So hopefully this has given you some great insight into what it actually takes to identify some infectious agents and helping people to get better. Because the first thing that you need to do prior to starting a treatment is determine what is actually affecting somebody that is ill. So while there is a lot here, used in tandem, a lot of these techniques can be very effective for a timely identification, which can help people get back on their feet a lot faster. I'll leave it there for now, but as always, if you do have any questions, please let me know at laura.nipissingU.ca. Have a great rest of your day and take care until next time. Bye-bye.